Greetings to my lecturer, Dr. Effendi. My name is Putri Nabila binti Sofian 106080. Today I am going to present my mock final year project proposal entitled Efficiency of Seed Biopriming with Trichoderma aspergillum syncoms on the growth and disease resistance of black pepper in Sarawak. As you can see, black pepper is a critical cash crop for smallholder farmers here in Sarawak, particularly in regions like Kuching, Samarahan, and Serian. However, excessive use of chemical pesticides affect health, livestock, and the environment. To address this, my project focused on a proactive biological solution, which is seed biopriming. Instead of using a single microbial strain, I am proposing the use of synthetic communities of Trichoderma aspergillum to mimic natural microbiome synergies. So, this research is actually needed because Sarawak's pepper industry is facing up to a 45% yield loss due to soil-borne pathogen like Phytophthora wilt. Currently, beneficial microbes are applied reactively after infection occurs. There is a distinct research gap regarding the proactive use of formula Trichoderma aspergillum syncoms for Piper nigrum. So, as you can see in my slide, my project aims to achieve three main objectives. First, to design and formulate a stable Trichoderma aspergillum synthetic community based on the synergistic and antagonistic compatibility between individual strands in vitro. Second, to evaluate the effectiveness of Trichoderma aspergillum syncoms formulate through seed biopriming on the resistance of seedlings, growth parameters, and physiological response, such as chlorophyll content of Piper nigrum. Plus, to determine the disease suppression ability of biopreprocessed Trichoderma aspergillum syncoms against major soil-borne pathogen, which is Phytophthora or Fusarium, under controlled nursery condition. My first literature review is black pepper and its agricultural challenges in Sarawak. Piper nigrum, also known as King of Spice, it dominates the international spice market and major source of income for smallholder farmers in Sarawak. Malaysia is one of the world's biggest producer. Production and export about 7,000 metric tons of black pepper in value 153.75 million ringgit Malaysia in 2021. Sarawak black pepper is granted as geographical indication due to premium quality influenced by factors like latitude, rainfall distribution, and ideal soil topography. Sarawak is one of the largest main agricultural export commodity, which is covering an area of 16,021 hectares with a production of 34,294 tons. Malaysia's black pepper production rise to 30,804 tons in 2020, up from 24,227 tons in 2008. Pepper yield increased from 4.4 tons to 6.4 tons per hectare. The sustainability of the black pepper industry in Sarawak is threatened by various soil-borne diseases, which is fungal and omnisete disease. The most destructive disease is basal rot or quick wilt, caused by aggressive pathogen Phytophthora capsicii leonian. Pepper plants at all stages of growth are very easily infected, attack on roots and the base of the stem, often causing sudden death in infected plants. Several major diseases in Sarawak is Fusarium wilt disease occurs in the main cultivation areas of Piper nigrum, affected the yield and quality of pepper. Second, yellowing disease caused by Fusarium solani. Third, blackberry disease caused by Cholatotrichum gliosporioides. Constraints of chemical control and the need for biopriming practices. The management of black pepper disease in Sarawak still relies heavily on the use of fungicides and synthetic chemical fertilizer. The effect of excessive use of chemical can bring serious negative impacts on the environment, human health, and disturb the ecological balance of the soil. The use of beneficial microbes often fails because usually applied after the pathogen infection rate on the farm is already too high. Seed biopriming technique is proactive by soaking seeds in a controlled manner along with microbial inoculation before planting. The function is for accelerate and standardized seed germination by allows beneficial microbes to establish early in young plant tissue to stimulate induce systematic resistance against soil-borne pathogens. Lastly, the concept of synthetic communities and nutrient use efficiency. Conventional biological control approach relying on a single microbial strain is usually less effective in the field. It does not have synergistic resistance against multiple complex pathogens. So the alternative is synthetic community design by combining several beneficial microbial strains to mimic the synergistic interaction of the natural microbiome. Stability of syncom structure and function completely depends on the support dynamics among its members. The loss of even a single critical species can alter the community composition and compromise its effectiveness. So moving on to methodology, which is the core of this proposal. First, Trichoderma asperorum isolate will be cultured on potato dextrose agar for 7 days at 28 plus minus 2 degrees Celsius. Spores will be harvested using sterile distilled water containing 0.1% when 20, and the concentration will be standardized to 1 times 10 to the power of 7 spores per milliliter using a hemocytometer under a microscope. So next, to construct the syncom, a dual culture compatibility test will be performed on PDA plates. Only compatible strands that show zero inhibition zones against one another will be selected and mixed at a 1:1 volume ratio to create the final syncom formulation. Next, for the plant material, healthy black pepper seeds will be surface sterilized with 1% sodium hypochlorite for 3 minutes and air dry under a lamina airflow cabinet. The seeds will then be bioprimed by soaking them in the syncom suspension for 12 hours at room temperature to initiate early microbial colonization. So next, to test disease resistant, target pathogens like Phytophthora capsicum or Fusarium solani will be cultured, and a standardized concentration of pathogen inoculum will be introduced directly into the polybag soil medium to simulate disease pressure in the greenhouse. The greenhouse experiment will follow a completely randomized design consisting of 4 treatment groups. First, T1 with negative control, seeds receive water only with no pathogen to monitor normal growth. T2, positive control, seeds receive water only but face pathogen stress to measure raw disease severity. Third, T3, syncom treatment. Seeds are primed with syncom with no pathogen to assess plant growth promoting traits. Last, with full biocontrol. Seeds are primed with syncom and challenged with the pathogen to evaluate overall biocontrol efficiency. So, through this project, I expect to successfully develop a stable, compatible Trichoderma asperorum syncom formulation. This biopriming treatment is expected to deliver uniform germination, robust seedling growth, and highly effective suppression of soil-borne disease. Ultimately, this will provide vital scientific data on using Sarawak's native microbial resource as an eco-friendly alternative to chemical fungicides. This is my work schedule. The 10-month schedule outlines the progression of my research. And this is my reference. Thank you.